Microspectrophotometry. A microspectrophotometer, I know it's a mouthful, there's not a good abbreviation for it, is going to detect the wavelengths of light that are reflected or absorbed from an object. With a polarizing microscope, stereoscopic microscope, compound microscope, comparison microscope, you are looking at visible light wavelengths that can hit an object and bounce back to your eye for interpretation. This is going to be able to look at different wavelengths of light with a much higher degree of specificity. Every substance reflects light slightly differently. They might all look the same to you to the naked eye. If you have a nice green leaf, naturally I color it in with red, you have all wavelengths of visible light, all the colors hitting the green leaf. It reflects back, because it's actually green, it'll reflect back green and your eye will pick it up, and all the other colors in the rainbow will be absorbed into it. Not only that, but only specific wavelengths of green are reflected back. So some greens are reflected back, while others, perhaps less so or more weakly, or others are absorbed. If one green leaf looks this green to your naked eye, and you have another one on the same tree that looks like this, perhaps one of those leaves is healthier than the other. Perhaps one has a sort of a chance degree higher of more of chlorophyll. Perhaps one is hitting the light differently. They're going to have a very slightly different set of green wavelengths being reflected back. Our eyes will perceive them as the same color though. A microspectrophotometer can quantify the wavelengths of light that are reflected back from objects to a very high degree of specificity and characterize those wavelengths that are being absorbed as well. Oftentimes you'll see your data come out as what's called an absorbance spectrum. This is going to give you a higher degree of differentiation abilities than anything using regular old looking at it with your eyeballs. It's more specific than you can discern, and it has more quantifiable, and your data is quantified, it's less subjective. So your microspectrophotometer looks something like this. You have your light source that's going to come up from the bottom, just like before. It's going to get condensed, just like in a compound microscope. Your sample will sit here. You have an objective lens, just like usual, and then you've got a mirror back here with a hole in it. An aperture is a hole. So that image is mostly going to bounce back here, and you'll see that with your eyes. A little sample of that light, though, it's representative of the whole object, but a little bit of that light is going to come through and get analyzed up here. What'll happen up there is shown in the diagram at right. That beam of light will travel up and hit a mirror. It'll hit a mirror with a prism here, and that will splay out the different wavelengths of light that were reflected from this object in the first place. Using another mirror, those wavelengths will be focused and detected over here with a computerized detector. This will be fed into the computer. So you will see a whole image through here, just like a regular compound microscope. A little bit of the light will be sent up here though. The different wavelengths of light will be split out, like when you send light through a sun catcher or a prism. And what wavelengths of light specifically were able to be reflected and spread out in that prism, because not all of them would. Remember, some get absorbed into the object and only some get reflected. Those that get reflected can be characterized by the computer. So this is what microspectrophotometry data looks like. This is a really useful technology to employ in characterizing paints. And the PDQ, as we had mentioned in the database slides, uses this data, this reflectivity and absorbance data from microspectrophotometry to characterize different types of paint that different manufacturers have. So here are Like here, this is a Honda from a Marysville plant in 2009, from 2013, from 2008, from 2006, and this is a clear coat. Now all of this is just going to look clear to your naked eye, but when you do this process, this microspectrophotometry process, the computer can characterize the different patterns of wavelengths of light. They can be subtle. You can see differences here pretty easily, but looking at the differences in this range of data with your naked eye, the difference between each of them is not that obvious. This data makes clear what the differences are, and this can be spat out too as a report with numbers on it instead of just these wavelengths. When you have a car that's in an accident, a hit and run accident, it left some of its paint on the thing it hit before it drove away. Certainly some of the clear coat. The clear coat's the outside most clear coat. If you run that through just a regular compound microscope, all of it's going to look the same. If you run it through this clear coat, the clear coat through a microspectrophotometer, you can tell whether it's the clear coat that Honda uses, whether it comes from the Marysville plant or whether it'll come from the Coatesville plant or the Shenzhen plant or the whatever, what year it is, etc. You can get a great deal of class characteristic information out of this process. Major benefits, again, it's a modified compound microscope, so the technology isn't that uncomfortable or new to use in terms of just handling it, manipulating it. You do need to have some extra training, though, to really understand the data that comes out of it on the computer end of things. It'll display the image on a computer too, so oftentimes there's a camera built in so you don't have to use the eyepiece. You can look at it on a much larger computer screen, which is helpful and convenient. It can detect properties of matter that other forms of microscopy cannot, and your eyes simply cannot discern. It quantifies the differences in wavelength to high specificity, so there's more objectivity in determining the class characteristics of this evidence. A human being is still going to look at the numbers and look at those patterns, the wavelength data, and make a decision. But because you have a higher degree of objective data in the first place, the person who's making the final judgment call effectively has to do less subjective work to get to a conclusion. Drawbacks, it is pricier than the other forms of microscopy. Outrageously useful though, so it's often something that a department will decide that this is worth having. It does require a fair bit of training to use and interpret the data, but that, otherwise just looking through the eyepiece, if you can use a compound microscope, you can probably use this. And because it is a very highly sensitive and sophisticated piece of equipment, it requires specialized maintenance, which can be expensive. Not all departments will want to spend that money.